Hi, I'm Ryan Felice. I'm a lecturer at University College London, and I'm really excited to share with you some of the key points of my presentation on the evolution of skull morphology in crocodiles, alligators, and their extinct relatives. So we're really interested in understanding the macroevolutionary dynamics that shape anatomical variation in crocodiles and their relatives, because these animals often get a bad reputation as being living fossils, partially because this lineage is very long-lived, but partially because the extant forms are relatively homogeneous in their anatomy and ecology. Largely, essentially all forms today are semi-aquatic generalist predators. But that wasn't always the case. There was lots of ecological and morphological variation in this clade, especially in the Mesozoic. So there's been a lot of research trying to understand how shape variation has evolved in these animals, but it's largely looked at quantifying just the outline of the snout using geometric morphometrics, and then concluded that the most variable thing about crocodile skulls is the elongation of the snout. But what if we used more complex methods to quantify the shape of the skull more fully? What could we then learn about how shape variation has evolved in these animals and whether they are in fact living fossils? So we used a technique to quantify the entire surface of the skull where we used 3D geometric morphometrics and using a semi-automated procedure project landmarks over the entire surface of the skull, giving us a very in-depth quantification of skull anatomy. We did this using 44 species of extinct and extant Crocodyliformes. We explored the morphological variation using some ordination techniques, and we also used evolutionary modeling in Bayes traits to look at the rate of evolution through time across the clade. Some of the key points of morphological variation in our sample: indeed, snout elongation is an important part of skull variation in this clade. PC axis 1 defines whether you have a long snout or a short snout, and it separates our aquatic piscivorous taxa here from our short-snouted terrestrial herbivores here, and the semi-aquatic generalists right in the middle. PC axis 2 defines some other features of the skull that wouldn't be captured if we were just using more simple 2D morphometrics. We can see that it has a lot to do with the orientation of the orbit, whether the orbit is on the dorsal surface or the lateral surface of the skull, and whether this structure here, the pterygoid flange, is large or small, dorsally laterally oriented. This structure, the pterygoid flange, you can see it pretty well here on this skull, is an important part of the jaw joint in these animals. It essentially forms an open buttress joint with the mandible and is thought to be related to resisting reverse wishboning during extreme bite forces. So it's related to dietary niche evolution in these animals. And so it's a really important structure that it seems highly variable, that we would not have known about how variable it was if we never measured the 3D anatomy. We separated out each different bone or region of the skull and looked at the variation there. And again, you can see there's a lot of variation in the dorsoventral features of many of these elements, especially that pterygoid here, the occipital region here, and some other regions such as the maxilla, for example. Taxa that sit low on PC1 have very tall maxillae, and taxa that are high on PC1 have very dorsoventrally compressed maxillae. So this is showing us that it's really important to capture the anatomy of these animals in 3D. When we look at the rate of evolution through time, on this figure, hot colors represent fast rates of evolution, cool colors represent slow rates of evolution. No matter what phylogenetic hypothesis we use, we get really fast rates of evolution in crown crocodiles, in particular this clade here, which represents a monophyletic group of crocodiles from Southeast Asia and Australia, which shows us that despite being superficially homogeneous in their anatomy, there is actually rapid evolutionary change in these recently diverged animals. this is telling us that, in fact, crocodiles are not living fossils, but have a dynamic evolutionary history, and to this day continue to evolve quite rapidly, even in comparison to some of their more unusual extinct relatives. I'll wrap up there, and I hope that I'll see you at the Q&A. Please don't hesitate to reach out to me at my email or Twitter to ask more questions, and thanks very much.